Hi everyone, welcome to lecture 11. Really nice to see you in our course on systems aging, aging, aging here and there, aging everywhere. Aging is a song that is universally sung. Aging is a thing you can ignore when you are young. But when you are older and begin to have dementia, you'll think back upon this course and wish you'd paid attention. Aging, aging, aging here and there. Aging everywhere. Aging everywhere. Aging. Woo-hoo. So today, very exciting topic, long life in a pill. And we'll talk about, first, before the break, about extending the healthy years we have. We don't want to extend life and just find out we have more years of disability. So how to compress morbidity. And in the second part, we'll talk about much more science fiction-y ideas about how to extend maximum human lifespan. How far can you extend it? From the more scientific to the more long shot ideas. All right, so we did welcome, song. Now we take a nice deep sigh of relief. We did that too. So, it's getting close to the end. We have just one more lecture. So if our course was a circle, we started off with, the patterns of aging, like exponential rise in risk of death, risk of disease, linear decline of function with age. Then we talk about the molecular hallmarks. Remember, we talked about mitochondrial dysfunction, chromatin modification. The first aging hallmark my teacher taught to me is chromosomal instability. You remember? That was a long time ago. Yeah. And then we looked at the saturating removal model of aging with its trucks, houses, the role of noise and stochasticity in aging. Today we're going to use it to understand strategies to extend lifespan, compress span. So with us. After that, we started talking about age-related diseases, the four horsemen: cancer, heart disease, diabetes, neurodegeneration, and why they're all rise 3 to 8%, reach a peak, and then drop. Explain all of them with one model, crossing a threshold, disease threshold. That's important for today's lecture. You have your damage, crosses the disease threshold. If you're susceptible, it pushes a parameter in a system. Like beta cells can't compensate into a regime where you're sick. It's like a threshold crossing. So we talked about the four horsemen. Then we talked about the legs of the elephant of health, right? We looked first at the first two legs. Remember the first one? Exercise and longevity, and how to combat the linear decline in function of, let's say, VO2 max and all these things. Exercise goes to 0 at 110. You remember that? And then we talked about nutrition and what sets the weight set point. How can you, why is it that we can eat 1 million calories a year and basically stay the same weight, remember? So, if you want to know the answer and you have a curious mind, let me take you by the hand and walk you through the leptin circuit. I'll show you something that'll help you change your understand. So we looked at nutrition. Basically, we talked about that every extra 150 minutes per week of exercise gets you, so first one gets you, let's say, a year of life, then a third of a year of life. You remember that? Or I don't remember exactly. Nutrition, much less known, but eat balanced and keep your proteins. Fine. Then we went to talk about, what did we do after nutrition? Remember? Yeah, we did sleep and stress. But before that, we looked at the genetics of aging. So what's different, what genes people who live to 100 years have, genes that give you diseases where you rapid aging, they're all in DNA repair, whereas centenarians have protective genes for, centenarians have X-critical genes, and progeria has houses, much faster building of houses, in specific DNA repair mutations, so very different. It's different to speed up aging and to kind of prevent early death. We talked about the genetics, and then we talked about the drivers of aging in yeast, and why we think they go all the way to human, which is two pathways, one in mitochondria, probably in neurons, and the other one in chromatin in telomeres, which is probably in dividing cells. Those two pathways, we have to address at least two different pathways of aging. And then we talked about the other two legs. Sleep, no, yeah.
It was 305 before.
What was it? Oh yeah, right, you're right. We talked about aging across the tree of life, of whales and bats, and how evolution tunes aging, primarily by changing ETA, and what we can learn from long-lived animals. And there was actually a talk. Did anybody here go to the talk by Chaim Cohen this week? So he talked about, so in the lab we can change, with the one mutation we can change, let's say, mouse lifespan by 40%, most. But evolution can go from mouse 2 years to bowhead whale by 100-fold, increase lifespan by 100-fold, in just probably like order of 10 million generations. Because the divergence of mammals, pretty quickly, let's say 10 million years, you got to a cow, whale, bat. It's like incredible plasticity of the mammalian radiation after the last extinction 65 million years ago. Because you had some mouse, the rabbit thing, now it had free niches. So this bone grew and you can become a bat. And like this, you can become a whale. So it's amazing. Also lifespan. So evolution can do something dramatic to lifespan. And so Chaim Cohen looked at not DNA changes, which there are too many, there's billions of them. He looked at something that's much smaller, it's still 20,000. It's places where proteins can be modified by adding acetyl groups. It's called acetylation. It's kind of can activate and reduce proteins. There's only 20,000 of them, so you can compare it between 100 mammals. And if you change the lysine to another amino acid, it's like acetylation, and there's another amino acid where it's like deacetylation. So you can see in the genome where it's fixed, like the regulation is activating always or inactivating always, or open to regulation. And comparing that to long-lived animals and short-lived mammals, and he found this very, very coherent set of changes, which, by the way, match this yeast thing. Just so the systems are just the same, basically, as what we found in yeast. But the upshot is that now you can imagine that there's 200 changes that will, quote unquote, make a mouse live like a whale. Like if you follow this line of reasoning. So again, one of those crazy ideas is now that it's possible to synthesize genomes, to try things like that. But instead of one mutation, you need to do, let's say, several hundred changes. Of course, there's also phosphorylations there, but maybe it's a finite problem to play like evolution does. Because evolution doesn't do one gene at a time. That kind of blew me away, that just to think about that, just looking at comparative, the tree of life and the changes, and figure out how evolution did it on a... Did I explain myself? Okay, so that was very interesting. And then we looked at at what? At sleep and friends and stress, right? And friends and wisdom, things that get better with age, right? The wise cognition, emotional regulation, decision making based on experience, balance, values. Remember? So talked about the possibility of having a so the way people think about the future of human population is that since 1900, different countries at different rates go through what's called demographic transition. Did anybody ever hear of that? Yeah. What is it?
The birth rate goes down.
Yeah. So it's basically women get educated, birth rate goes down. It happened, and they think this is going to limit human population to 11 billion around 2100, with another billion people in Asia and another billion people in Africa. You believe that?
I think 10.3 is
10.3? Okay. So, once the population is at steady state, each person who's born, somebody dies. Each person who dies, somebody's born. What happens if you extend the lifespan? Now, at time t equals 0, I extend the lifespan by 5 years. What's going to happen? So first of all, you'll get a transient increase and then go back. And it'll just be that the population now will include not only people who live to 90, people who live to 100. Just the age distribution will be uniform. You'll have each person who's 2 going to go 3, another person replaces them at 2. So the fraction of people at each age will be constant. What you increase is the number of old people.
Wait, we go up and down? Why not just up and stay that way?
Yeah, maybe it'll go up and stay. I don't know. I can't calculate right now. What?
Depends on what limits the population. If the limitation is, for example, the energy that we can get from Earth, then
Yeah.
the amount of food we can
Yeah, good point. This is like maybe I got into like too deep water for me. Let's go back to the shallow water where I'm comfortable. Alzheimer, right. So we talked about Alzheimer. So actually, this lecture was built, you went to the Valley of Despair, Alzheimer. Then we went to like a little bit optimistic, like stress reduction, friends. And then we got to the wisdom part. Got to the wisdom part. We looked around. And now we're going to look to the future and talk about the cutting edge strategies that are now happening to extend lifespan and maybe try to extend health span, expand maximum human lifespan. Right? That's what we're going to do today. You ready? Let's take a nice deep sigh of relief. The question is if I'm ready. Only one way to find out. All right. So the concept of health span. So if we can think about a human life, typically we're reasonably good health, and then at old age start chronic illness, disability, and functional decline. Of course, that's just a simplification because you could be ill, well. Let's imagine, it's just for simplicity, think about the lifespan as having, let's say, a health span and a sick span. Right? This is the 6 span. And in 1980, an influential writer called Fries said that considered possibilities. What happens if we extend lifespan? So one depressing possibility is this, right? This is lifespan extension purely by extending the 6 span. And I don't think a lot of people want this. Maybe if I extended here by a certain amount, let's say by 10%, I can also extend the health span by 10%. And I have like what's called scaling or proportional. So that's one possibility, right? It's like stretching time like a rubber band. What I want to tell you is that it's so important, but the state of the field now is that there isn't data on what actually happens when you... Almost. I'll show you the little data that there is about this health span. It's hard to measure. You have to measure an animal over time. In humans, there's much more data about this. And I'll tell you what happens in human population. You can also imagine compressing 6 span. Or as Fries said, call it compressing morbidity. Morbidity means illness. So you extend lifespan by extending the health span. Or even by extending the health span and shrinking the absolute 6 span. And he said like the ideal life you want is you're healthy, and then you die without being sick. We're very short 6 span. Actually, centenarians, about half of them are like that. They reach 100. As I told you, but half of them don't have the 4 horsemen, and they basically are sick very short part of their life. For sure, relative, but also absolute. So it's possible to do. And it's like a righteous person's death, right? You go to sleep and don't wake up or whatever. So I want to talk about this compressing 6 span as a kind of ideal for the neuroscience field. And how to do it. And we talked about, of course, reasons to do it is to stop suffering, to gain the productivity and reduce the healthcare, the wisdom of these people. Some people say, but that'll grow the Earth population, but it's revealing that nobody says that about curing cancer. No, it'll grow the Earth population. Nobody says that. Here, you're actually delaying hundreds of age-related diseases, including cancer. So this is how to do it, right? So you need to follow health. So a lot of interventions that increase lifespan in animals, what you do is you measure at a certain time point their health, and you say, these are the control animals, these are the extended animals, and look, the extended animals are healthier, right? But that doesn't tell you necessarily which one it is. It's the one time point. That's like standard field. So a lot of like interventions that extend lifespan in worms, flies, mice, I'll tell you about, improve health. No doubt about it. So the mice run on the wheel, like even though they're like 2 years old, maybe like, I don't know, 70-year-old people, but they run on the wheel like young mice, more or less. So, but what's the proportion? You need longitudinal studies. Those are coming. There's like a longitudinal study of mouse aging, but it's going to take a few more years to publish. Okay, so how can we approach this? And so what I'm going to tell you is, I'll show you, there are certain drugs right now which are FDA approved and used for diseases in humans like diabetes. And there's good reason to think that they might slow down aging because they not only, when you look at the people with diabetes who take them, they have less cancer, less Alzheimer's, less horsemen. Okay? and they extend life in mice, etc., etc. The thing is, we want to prioritize them or to know whether they're going to scale the lifespan or increase this span or compress it. We want to know which drug to do an expensive clinical trial. And I'll tell you about clinical trials that are already actually going on. This is very new because I would say the first real clinical trial, a small one, was published August 2024 on aging. So it's very new that you can do that. But it's going to be very, very, very huge, I think, field. Okay, so and next time, the last lecture will be open questions in the field. Right, but today I want to focus on drugs, things like that. Okay. And so that's a review. So we talked about compression of morbidity. So let's see how we can of course in this course the special thing is we have our mathematical framework. Okay, so we have our SR model. And you remember that you have damage. The simplifying assumption is there's a form of damage that drives death, disease, and decline. And then we can purify it in a test tube and understand what it is. Right? And if you look at a certain individual, it goes like this. And when you cross the threshold in the model, you die. And here's another individual. So that's why the death times are different, and they're distributed. The risk of death goes exponentially with age, decline is linear, all the things, all the patterns we get from the SR model. We know we have a good idea what this X is in mammals, systemic senescent cell load that causes inflammation, and another one in the neurons that we talked about, lysosome, deacidification, mitochondrial dysfunction, protein aggregates, all this stuff. How can we do six-span? So in one of the lectures when we talked about the horsemen, we said that people susceptible to disease have a lower threshold here, and when they cross it, they get sick. Not everybody's susceptible to each disease, but these two people happen to be susceptible to the same disease. So this one's going to get it here, and this one's going to get it here. You notice it's before they die, of course. Because disease you get after death, we're not discussing in this course. Only one. So this time difference, this is the six-span of this individual, of this individual. Did I explain myself? So we have a mathematical way to address. Now, in order to address the six-span, let's say we saw there's hundreds of age-related diseases. Each one has its own threshold. If death threshold is 17, they can have 12, 13, 14. Let's put them all together at an average threshold, let's say 15. According to everybody's going to get something. And call that the six-span. Just to simplify things. Each person, we need an individualized model like you're building for each person. What are the thresholds so we can predict? Let's just make an assumption. There's an average six threshold. Now we can calculate. And what I want to do is say these drugs, what do? They affect something. Houses, trucks, threshold, noise maybe. So which of these parameters scales the six-span? Which of it compresses it? That's what I want to know. So you can calculate that because we know the solution for the average damage, and we can find what time you die, what time you get the disease, and subtract them. And I'm going to normalize by the time you die. So this is going to be the relative, so what percent of your life is six-span, and you get some equation that has all the parameters. Doesn't matter. You have an equation. And from this, you can see what's going to happen to the six-span. And the conclusions are that if you affect the houses, you basically stretch time like a rubber band. And you get that basically you extend the absolute six-span in years, which is bad. And you keep the same relative six-span because you extend lifespan and six-span at the same time. Relative six-span. And the sign of this, as you recall, is that the survival curve scales. Survival curve has the same shape. When you normalize time by the 50% time, we did this many times. So scaling survival curves, which are much more commonly measured, means it's bad for six-span. So now we have a way to be careful of these drugs. If we have a drug that increases lifespan in mice, let's say, but doesn't change the shape, it stretches time, it's going to stretch the six-span. On the other hand, changing any of these other things gives you, as you recall, a steepening, compresses the six-span. So here we have a criterion. We can look at an intervention, and there's many. Look at the survival curve, there's hundreds, and prioritize. If it's scaling, stretch time like a rubber band. Bad. If it's steepening, it doesn't extend lifespan by that much, but it's going to compress the six-span. Good. So we have a prediction, a tool to use. So what I want to show you is what drugs, according to this, are the best candidates right now for. So if you're a longevity doctor, so there's actually in the last few years, a new medical specialization, longevity medicine. Do you know about that? You heard? You have it in your hospital? You're working on it? Far away. Because it exists in Sheba. So you guys better start. So anyway, it's really good for making money for doctors. You take healthy people, and their job is to tune your nutrition, exercise, sleep, friends, and the drugs I'll tell you, to make sure that you're like an athlete for longevity. That's their meshugas. That's what they want to do. Yeah.
If we'll take both drugs, so we are both living longer, but
Yeah, combinations.
yes.
And we're going to get to that. They're good. Yeah.
Do we have observations on Olympians? Because also the slope was.
Yeah. So I talked about Olympians. Yeah. So Olympians they live a little longer and they have a steeper slope. And because what happens for them is they have a higher X critical, and they have a shorter six-span. Yeah, that's right. Yeah. Yeah. Good point.
You increase X critical and not X critical.
Yeah, good point. There's one you found, because you're a mathematician, you found the exception. You're right. I think that's what you're going to ask. If you just what you said is, if you increase only X critical and not X disease, that's bad. So that's like imagine an imaginary experiment. We take sick people, they're sick, and you put them in an emergency room or like a intensive care, and you keep them alive. How? You increase their X critical. You give them oxygen, and then you pull out the plug, and they all die at the same time. That's very steep, right? But their six-span is very long. So the one exception is if you increase X critical, but keep XD the same, like with your intensive care unit, and now all the six-spans are longer, but you can make the survival curve steeper. Is that what you meant?
Yeah.
You get a gold star. So that's the one exception, so you have to be careful about that. But we think that's very artificial, because if under the assumption that the same driver causes both disease and death, you have to do something special to increase X critical and not X disease. But be careful of this intensive care unit. Make sure your parents sign a document saying, if I don't want to live, disconnect me. Okay, because you can do that. Advance directive. If you want to die with dignity, it's possible. Not everybody wants to do that, but anyway, that's okay. So now let's. And I just want to say, I'm going to gloss over it, but just to say that there are 4 studies that did measure longitudinal health span, let's see, the health of individuals, while giving them an intervention that extend their life. And now you can test this. And there's one paper in flies, there's 2 in worms and 1 in mice. So mice, you measure the frailty index, and they have different. You say they're frail if they have more than 4 out of 8 categories or something. And it works that in all these organisms, if you here I measure the steepness relative to wild type, and here it's the 6 span relative to wild type, you get kind of the predicted slope where scaling doesn't change the relative lifespan, which means stretching time like a rubber band. So this is in worms, and you can find this in the lecture notes and in this paper. Okay, on bioarchive. So it appears to work, at least in animals. And now we can take the literature and look for drugs, like these chemicals. If you can tell me what they are, you can get an extra point. This one you can see is an organic thing made by a plant. So simple, elegant. This one with this floor, you can tell it's not made by an organism other than humans. These are 2 very prominent possible pills for extending health span. But we think one of them is going to be bad, stretched. The other one is going to be compressed. Now we know. Not Ozempic. Ozempic is an antibody, right? No, it's not an antibody. It's a GLP-1 receptor. It's not Ozempic, but good guess. Rapamycin, yeah. Rapamycin. No, it's not rapamycin. Rapamycin is very, very complicated. It's like much more complicated. It's like it comes back to bite you.
Coffee.
It's not caffeine. I love it, you guys. You see, for me, this is like hieroglyphics. I don't understand these hexagons, but apparently they're carbons. This is a fluorine. Okay, great. I'm glad that you're interested. And now let's look at. Let's erase. I'm always like at a loss what to erase because. Let's erase this part here. And I want to tell you about a really very, very powerful experiment that's powerful partly because in this field there's a lot of companies. Sometimes companies lie, and generally not to be believed, their publications in this field. That's a broad statement, but there's a public effort from the National Institute of Aging called the Intervention Testing Program. It's been running for a long time, and they test drugs and supplements which are FDA approved on humans that people think for different reasons might be anti-aging drugs. And they test them in mice, but they do it very rigorously. First of all, they don't use black 6 mice like most of Weizmann. They use, because that's one genotype, so you can't ever know if it's that specific mice. They use outbred mice, which are genetically diverse. Second, they don't depend on one animal house facility. They do it in 3 different places in America. Third, they use hundreds of mice. So different groups have to get the same answer. And they do it standard, always the same. And they have enough budget, $3 million a year, to do about 7 a year. and they've tested something like 50, and now they have around 10 hits that extend the lifespan. Right? And they publish the survival curves. So we can look at the survival curves and see which are steepening, which are scaling. Which one, when you divide time by the median lifespan, collapses the same shape. We know that'll be bad for our purpose of compressing the sick span. So this is the median lifespan divided by wild type. So all of these extend life, and this is the steepness divided by wild type. So again, what we're talking about is the survival curve with the intervention. And steepness is basically the mean divided by the standard deviation of the lifespans. Steepness. Okay. So, I just want to say, they made a really important discovery by accident. They started with rapamycin. Rapamycin extends life in yeast, worms, flies, really great candidate. It's a molecule that's an immunosuppressant for transplantation, made by a fungus to stop the growth of other bacteria and stuff like that. And it's kind of mTOR inhibitor. It's like a starvation mimic, and it slows down the rate of life. You can see. And they gave this to mice. And after 22 years almost, they realized, or after a year, that they made a mistake. And the pills they gave the mice in their food, the digestive system broke down the rapamycin. It didn't reach their blood. And they said, oh God. So we have like 20-month-old mice. Let's abandon the experiment. And the leader of this, called Rich Miller, said, according to his testimony, no. Even though there's no chance to extend life to these old 20-month-old mice that are like 60-year-old, 70-year-old people, let's make a formulation that won't be digested and give it to them. And they did that. They knew it's a waste of time because they're already like this. And amazingly enough, it extended their lifespan. So there was the first demonstration in mammals that you can extend lifespan at old age. And that's why in 2009, that paper is like a big, important step. But it came from a mistake. So where's rapamycin? It's here, which means it's scaling. Just like caloric restriction is also scaling, it's like here. So it's going to extend the sick span. So we can already tell you, or maybe to answer your question over there with the laptop, you need to add something to this. Right, so
Those two drugs together should do it, no?
this plus this.
Yeah.
Yeah, that's right. So then there's a lot of other things here. There's aspirin, metformin, which is a very common diabetes drug that's made by a plant, or used since 1920, safe in people. So people, the first drug advocated for a big clinical trial on aging, the TAME trial, was with metformin. Nir Barzilai from Einstein pleaded with the FDA to approve a clinical trial on aging, because aging is not a disease. They don't allow clinical. And then they formulated a way to do that by actually measuring many, many age-related phenotypes. So that's like aging. That trial did not get off the ground yet, but it inspired a lot of trials. And there's a trial, rapamycin, there is a trial in humans. It was published very recently. It was small, 60-year-old, healthy people. And they found, after 48 weeks, beneficial effects. In women, more muscle mass, and in men, more bone mineral density. it's not powered enough to tell you a lot, but it's a step. It's a first, actually. Placebo, random. So some people got placebo. They actually had more side effects, including mouth sores, which are class. The placebo had more. And very low, like 10 milligrams per week, very low dose. But it's like a first, okay? You can do it. And again, a year ago, 2 years ago, it was like a taboo. You can't do clinical trials on aging. Now, there's a lot of them in some countries like Singapore, Japan, where aging populations are massively investing in that. So all the drugs, I'll tell you, they have clinical trials. So, okay. Yeah, it's a big change. Like, this field is like this, like, in terms of every parameter you can think of. It's like, who knows where it'll reach? Yeah.
Why was that taboo?
Because aging is not a disease.
It's like a technicality, or
I think it's a serious mindset. So culture is very interesting. But once there's a change, it's like opens the mind. So, all right. Now, what's up here? Because we want steep, right? And lifespan. What's up here? In order to, so what's up here is several classes of medications. One of them is senolytics that kill senescent cells. So they kill the X here. There's a bunch which are basically diabetes drugs. SGLT2 inhibitor, acarbose, ketogenic diet, and ketogenic diet mimetics, antioxidant, green tea extract. It's lovely, right? And then you have some kind of blood pressure lowering drug. These guys are like dose dependent, and you can start them at old age. Dose dependent move like this. So we think they're good candidates for compressing the lifespan. Now, what happens when you add one of these steepening with rapamycin? So if you think about them, these act on these parameters. Rapamycin acts on this parameter, so they should be additive because you're affecting two different parameters in the body. If it was two things affecting the houses, you don't know how they add up. And when you take, like, say, acarbose plus rapamycin, it's like adding vectors. This vector and this vector. This is the answer. So you get more long-lived and steep. Isn't that cool? That's what the model says. That's what the ITP result says. So combinations, certain combinations are also good. But of course, this won't get you to 200-year bowhead whale, right? We're talking here. And it's mouse. Remember, mouse is very different from human. We talked about those differences. I won't repeat them, but there's important differences in the SR model. So, all right. So what drug should we prioritize out of all of these? So Neil Barzilai also has a great list that I put in the lecture notes, where he takes all the suspects and he orders them according to evidence. Do they affect hallmarks of aging? 1 point for each hallmark of aging. Have they been tested in mice? 1 point for each mouse experiment. Have they shown in human epidemiological studies that they improve several age-related diseases, reduce the risk for cancer, Alzheimer's? The top of the list is SGLT2 inhibitor. I'll tell you what it is. Next one is metformin. The next one is, there's acarbose. There's the GLP-1, what's it called? The Ozempic. Okay, so there's a list. What's this nice guy here? Do you ever prescribe it? You don't? For gestational diabetes?
yeah, metformin, yes.
No, but SGLT2?
No.
No? Why?
It's not in our.
It's not in your toolbox. Yeah, it's kind of new, but so this is it. And I just want to tell you the wonder of biology right now. It works on it lowers glucose. How does it do it? It works on our kidney. The kidney job is to purify the blood. You want to throw out all the toxins from the blood. Right? So you think, let's make a pump to throw out each toxin. But that logically is a problem because you want to deal with toxins you've never seen before. Evolution has never seen. Maybe there's a new spider. Maybe you're living in the Haifa and you get some... I don't know. So the kidneys do something very brilliant. First, this is the drawer in the blood. They throw everything out, everything, except for proteins. And then they have a pump for what to keep. Glucose, salt. Right? It's brilliant. So this SGLT2 blocks the pump that brings back sugar. Okay, that's why it's a great diabetes drug. Also, you lose weight and stuff like that. But it does a lot of other things, including, turns out, kills senescent cells in some way like that by activating AMPK, reducing PDL1 checkpoint inhibitor, T cells kill this, something like that. It kills senescent cells. It does many, many things. And in general, I want to tell you that there's some common theme to these guys. And that is, they reduce sugar spikes. So you eat a meal, above 7 millimolar, damage to your blood vessels. That's why diabetes is at 7 millimolar. So these drugs, you can say one thing to do is protect your blood vessels. Yeah. And they're like trucks. What's the connection between blood vessels and trucks? Yeah. What in our thinking, the X is senescent cells. The trucks are immune cells that come and eat them. How did they get to the senescent cells? Through the road system, right? So when you have like poor capillaries, you can't have immune tissue infiltration. So there's an experiment done in Israel where they made mice that have a lot more capillaries for a different reason. Miriam Grunwald told me she forgot about them, and then they called them from the animal house after 4 years and said, "Do you want your mice?" Mice lived 2.5 years. She came. The mice were so just like, and then they repeated it. They did a paper, and increasing the vasculature makes them live. It's like world record. It's like 50% more. Very steep. Very. So this blood vessel is a key intervention point for long-lived steep. And these guys are like starvation analogs. They slow the rate of living, mTOR, they scale. So these are two different strategies, you can say, for mice lifetimes. In humans, blood vessels are even more important because mice don't get vascular dementia and all this stuff. All the brain really needs the vasculature, and we have our heart attacks that mice don't get it. So like in humans, even more vasculature, more important. So it's thought that this SGLT2 does many things, but these guys, ketogenic guys, reduce glucose spikes and thought to extend health, or candidates to extend health span. And now there's clinical trials in Germany for aging on SGLT2 and GLP-1 and combinations of them, et cetera, et cetera. So another important thing, sexual dimorphism. Male mice, female mice, they live longer. Who do you think, which sex, it's easier to extend life? Now everybody has to raise your hand. If you think male, raise your hand. If you think female, raise your hand. Okay, so it's not easy to know, but it's male. So most of the interventions, SGLT2, they work on males. The VEGF works on females, and green tea extract. Sorry, I'm gender fluid here. So it's green tea extract. All these other ones on males. So it's like different. Also, non-feminizing estradiol works here. Maybe also protects the blood vessels. The male blood vessels less protected than the female because they have estrogen in mice anyway. But there's a big sexual dimorphism, and we're going to need different drugs for males and females, according to the mice work. I think very interesting. It's like females are already pushed to the extreme of longevity because they're long-lived and steep, also in mice, also in humans, and we need extra ways to extend lifespan, but they exist. When I get a paper to review, I look for what is fresh and new and how to realize its full potential. Of course, I note what to improve, but I always let the authors choose. After all, they know their own work better than I do. Hallelujah, I'll review ya. Hallelujah, I'll review ya. Now you know I've been reviewed before, and I lay crushed upon the floor, but I won't pass on the aggression to ya. 'Cause science is a social affair, we help each other to get there, to find the truth and help it to shine through, yeah. Hallelujah, I'll review ya. Hallelujah, I'll review. Hallelujah, I'll review ya. Hallelujah, I'll review ya. Okay, so we talked about how to extend the health span by looking for steepening or combination of scaling and steepening mutations, and there's a list of FDA-approved drugs that are safe and have mechanisms across the hallmarks of aging, and clinical trials are beginning. Like Ido said, a polypharmaceutical that will address different aspects of aging, and maybe in the coming decades we'll have standard treatment for aging, or maybe personalized. I think that's pretty safe to say is coming. But what about extending maximum lifespan? You know, we live to 100, 110. Can we live to 100 and whatever? Some people promise 150, some people say 1,000. This is not to be believed, because we have no idea how to do it. In mice, you can extend lifespan by 40%, but it's much, much more difficult in human beings. And there's several ways to see that. One of them is that if you look throughout human history and you look at, let's say, religious order, meditators, nuns, etc., live very healthy lifestyle, social ties, sleep, regular meals, natural food, moderation, all the pillars of health, they don't live more than 100. They have more steep survival curves, but we would have someone who lived to 140, but we don't. Especially now, population growing. It's something very important for me to tell you, that population is growing, and the median lifespan is getting longer and longer. So if you look at 1800, 1900, 2000, median lifespan, it's going like this, and you can say that every day we gain 6 hours. Or in 200 years, we gained 50 years, 1/4. We start, let's say 80, 30. That's relentless. Yeah, there's wiggles and stuff like that. And now, by the way, in the US, for the first time, for a few years straight, median lifespan went down. And a lot because of young deaths, because of what's called deaths of despair. Opioid overdose, very easy to take opioids and stop breathing. No problem. Anyone can do it, not recommended. And fentanyl and all that stuff. And crime, inequality, etc. But that's pretty strong. Of course, it's not a mystery, just extrinsic mortality going down because of less. Used to be 1 in 100 childbirths was death of mother or child. Now it's like 1 in 1,000. Am I right? Yeah. That's big because if you die of childbirth, that reduces median lifespan, and etc. etc. But the maximum lifespan hasn't changed. It hasn't moved. It hasn't budged. It grows maybe logarithmically less, like maybe less fast and logarithmically than the human population. Very, very. It's not even exponential. It's like super exponential drop of survival. So, maximum lifespan is much harder nut to crack than median lifespan and health span. So, how do you do it? We need to redesign human biology somehow. And another piece of evidence comes from your teaching associate here, Ben Shinhal, who's been sending you emails, checking your exercises. Worried sick about you. Ben used different models like the SR model to ask how much variation in the population can there be in each of these parameters? Because we know that take twins, their lifespan is a little bit heritable. So that means some twins are in a genetic group that lives longer than others. They inherit some genes, and centenarians inherit genes. But which parameters change among people? It turns out that if certain parameters change by more than a few percent, you would have people that live to 140 because they affect lifespan so much. And those are houses and trucks. If you change them, you get people who live to 140, by more than a few percent. But you can change threshold and noise and get the heritability of lifespan. I mean, they can genetically vary, but they don't extend the maximum lifespan. So we think in human beings, the threshold changes, so that means resilience, also socioeconomic resilience. And noise, we don't know what it is, but change. But the nuts and bolts, the trucks and the houses, they just don't vary in the human population. As if they're like stuck at a certain value. You can ruin them. You can I told you about progeria. We have accelerated aging, DNA repair, errors, mutations, and then you have much more houses. So it's easy to live shorter, but not live longer. We need to redesign human biology somehow. It's a different problem. Maximum and median are different problems. Okay? So what is the cutting edge on maximum lifespan? Of course, for all of human history, people try to find the fountain of youth. Is it a fool's errand? What do you think? No. Yeah. You told me you don't think so. All right. So let's be fools and go into the cutting edge. So there's actually very interesting ideas and a lot of funding, a lot of companies, and I want to tell you it's very interesting. So let's erase this thing here. So one thing we can do is Okay, so let's put it this way. If we look at the SR model mechanisms, we know that we need to intervene right at the start, right? So we remember there's these histone acetylations. Here are the telomeres here. And there's the vacuoles and mitochondria and all the protein junk. Those are things that have to do with the ETA, with the houses. We want to slow down houses. Later we'll talk about increasing trucks, but slowing down houses is the most fundamental. So we need to clear those histone marks and get rid of the protein aggregate. But this actually happens all the time in human beings. There's immortality in human beings, and that happens in the sperm and the egg. So as I told you before, 40-year-old egg and a 40-year-old sperm fertilize with all their chromatin marks and the fertilized egg and the embryo is reset to age 0. The sperm and the egg get rid of those histone acetylations and those protein aggregates and those bad mitochondria and reset. Otherwise, life couldn't continue. We have a lineage going back to 1 billion years to the eukaryotic Adam and Eve, right? So, single cell ancestors. So, let's learn from the fresh eggs how they do it. And that's the idea for this biotech companies and some experiments that are kind of promising and intriguing. The trick is, though, you have to do it without erasing the identity of the cell. So you can't become an embryonic cell that has infinite potential to become muscle, heart, brain. No. We don't want to become a ball of shapeless cells, right? Most of us. Also, changes in histones are a step to cancer. So if you have these cancer cells that go back to this primordial stage, give you a terrible cancer called teratoma that builds teeth and hair and stuff inside your body. It's disgusting because it has this potential. That's teratoma. So you don't want to do this and get teratomas. You don't want to lose the cell memory. So you have to walk on a knife's edge to refresh the eggs without losing identity and without making cancer. Okay? And so what do the eggs do? So the sperm has a really neat trick. It just throws away all the histones and replaces them with a different kind of protein. It binds DNA, so you don't have a problem. Protamines. The egg uses these histone deacetylases to just clear the histone acetylations. They degrade a lot of their histones, build them back, new ones, and they do it. They also activate autophagy and clear the protein aggregates. They can do it. So if they can do it, we can do it, right? And so the approach here is to learn from the egg. It's called partial reprogramming. And the idea is based on the discovery that got the Nobel Prize that you can take a cell from an adult, add just 4 genes, these ones, OCT4, SOX2, KLF4, and MYC, and you become like a pluripotent stem cell that can be made into a neuron, a muscle cell. What's your favorite cell type? Sperm cell. So you can take an adult and bring it back to the embryo. So that's like the Nobel Prize. It's the basis for now research. So you take a neuron from somebody with bipolar disorder, or a skin cell from someone with bipolar disorder, change it to a stem cell, and make it into neurons, and find out what's different about their neurons. Did I explain myself? So it's amazing, right? You can take your cells and make new organs. Okay, But the idea is more bold, is to take a mouse and to activate this for a short amount of time. So that you don't go all the way back, but you go partially. Can you believe it? Take the mouse, and you do this for a short amount of time. And you also delete maybe MYC, which is an oncogene, makes cancer. So you only keep the transcription factors that turn up all the histone deacetylase, all that stuff. Right? So you use a part of the Yamanaka factors for a short amount of time. And you get kind of rejuvenation. So in mice, you get rejuvenation. There's a famous study where you had crushed optical nerve, and after this, the mice could see again. These kind of studies in recent years. And there's a very big company by Jeff Bezos that it's very secret. They took some of the best scientists, gave them very big salaries, and stopped them from publishing. And now they're trying, for example, all the possible combinations of 150 different transcription factors, and it's a secret, don't tell anyone. Okay? To go to town on this reprogramming. What do you think?
Not in the secret project. It's over, you can try.
Oh yeah, so there's a couple of papers with rejuvenation of mice you can look up in the lecture notes.
It's across all the cells of the mouse?
Yeah, so you I don't remember the technical details, but it's like you can pulse, you basically you genetically engineer a mouse and then you give it some drug that turns on the genetic cassette and you can determine how long and when you want to do it. So you have to so they try different schedules and it's very hard experiments. But of course the mice sometimes get cancer, so you shrink it. It's like finding the secret recipe.
Well, the neural connectivity, all the identity of the self, may change also the differentiation process.
You mean the identity of the mouse?
The brain structure.
Yeah. Actually, I have no idea. That's a very good question. Can you change the identity? Do you change the identity? The memories, right? Because in principle, what you want to do is pulse a wave will erase their bad histones, and stuff, but maybe they'll erase the identity. That's what you're saying. Great question. I don't know the answer, but I just want to tell you the cutting edge where people are thinking. So this is a big field right now, shrouded in secrecy because there's big money, unfortunately. And when you go to meetings about aging, you have to be careful to know which talk is from a company, which talk is from a real scientist, because it's very confusing otherwise. So that's one problem in a field like this, which has so much potential. Okay. What about increasing the trucks? Trucks are the immune system. You remember we said maybe the vasculature is like the road. The immune system can come to the senescent cells. So there's 3 different approaches at least to improve the trucks. It's far safer, I think. So one is drugs that kill senescent cells. These are the senescent cells. And the drugs, let's say, they drop a safe on them. And there's a big company called Unity again. It kind of did a lot of damage to the field by trying a senolytic that doesn't really work, called D plus Q for a long time. Clinical trials didn't work. Now they switched to senolytic that does work. How does it work? Zombies, they don't die, right? Why don't they die? Because their apoptosis program is blocked by this protein called BCL, which is anti-apoptotic. The senolytics block the blocker of cell death. It's an anti-cancer drug, actually. And then the senescent cells die because they depend on not dying Like they're very damaged and stuff like that, but they don't die like zombies. Now you allow them to die. They die. So there's this BCL2 inhibitor. The problem is, why is it not why don't we all take it? Because it causes low platelets. Because the platelets also don't die, also because of this BCL2. So you're like this. So what this company does now is inject it into a place where it won't go into the blood, like the eye, sorry about that, to treat glaucoma. And now it's in stage 2 clinical trials. Everybody's waiting. I think it probably won't work. You need a systemic reduction of senescent cells, but maybe it will work. I don't know. So that's one kind of drug. Now, if you think about the field of cancer, which is, I don't know, 50% of biology, all the money goes there. You try to kill cancer, it's so hard because the cells are evolving. They're different in each person. They're smart. They can become resistant. It's much easier to kill senescent cells. They're just sitting there and they're not dividing. They're not mutating. They're a sitting duck. So soon there'll be a senolytic. It's clear to me this is going to be a solved problem. The question is, well, how much will this buy us? I don't know. I'm very optimistic about that because it's so easy. It's so easy compared to cancer. It's like, wow. All right, so that's senolytics. Second is to improve our immune system. So again, taking senolytic is actually an anti-cancer drug. This is also anti-cancer. You take T cells from a person, and you genetically engineer them to have receptors that recognize cancer cells. That's a cancer therapy. Scott Lowe, a couple of years ago, did that, where the receptor now recognizes something on senescent cells. And in mice, you could clear senescent cells, rejuvenate. So that's another approach to design the immune system, T cells to kill the senescent cells. Did I explain myself? That was actually invented in Weizmann, the CAR T cell.
Aren't the immune cells already designed to kill senescent cells? Like why are the CAR T cells better?
Why are the CAR T cells better? Because, as I said, the immune cells that are designed are great at it, but then they get overloaded. So here you're adding a whole new kind of trucks that don't care about senescent cells. And yeah.
You also need senescent cells. Like if you just like kill all the senescent cells, if I'm injured, then like it will be a problem. So is it like irreversible? Do you add the CAR T cells and now like it will get cut?
No, if you get cut, you'll get new senescent cells.
And the CAR T cells won't.
Will they slow wound healing? It's a good question. I don't know about that.
My question was more specific. Does CAR T cells remains in your blood forever, or are they like you inject them and then they?
Yeah, they have a finite lifetime. They don't.
Okay.
Yeah. You can stop it. Yeah. Okay. And now, by the way, now with synthetic biology, there's labs who are making these intelligent T cells by putting in circuits made of transcription factors, proteins. They can compute. They say, okay, if we send this, but not this, exclusive OR sends this and this, then secrete a vesicle with RNA that puts a circuit into that cell, and it's like incredible, programming the minds of cells. That's like the cutting edge now mainly for cancer therapy. So cancer therapy, they have big weapons. The senescent cells are so easy. Okay, so and the third thing is again from cancer. The senescent cells have a "don't kill me" signal that cancer cells also express. So this inhibitor of this. So this was also discovered in Weizmann that if you inhibit this "don't kill me" signal, the immune system kills senescent cells better. So this is a very well-known cancer therapy. Moved to the field of senescent. So I think that part I'm really optimistic about. Great. So lyrics. Now I'm going to go now science fiction and more science fiction. People started talking about AI, and I was very skeptical. 2 years ago, he told me AI is going to help make new drugs, and I said, I'm not skeptical anymore because of the advances we all saw. So the idea is that AI accelerates new drugs. So if you have a target like this, right now making drugs is long and expensive. Takes 10 years, $1 billion, 9 out of 10 fail because mice are not equal to humans. What's the pipeline of drug discovery? You need a target. Then you need to find a molecule, peptide, antibody method, to bind it and do something to it. Then you need to see if it's toxic. It binds else to non-specific or like off-target, may causing side effects. Can it reach the body, the blood, etc., the blood-brain barrier? And then you need to do clinical trials. Phase 1, see if it's toxic in humans. Phase 2, is it effective in a bigger cohort? Phase 3, will it work on thousands of people at hundreds of millions of dollars? It's a long process, so that's why drugs are so difficult. But AI can help in all of these. One day it'll find targets. Right now, Nobel Prize this year was for AlphaFold. This problem, I was educated on this problem. So you have a chain of amino acids, they fold on itself, do the physics to find out how it folds. This AI software, AlphaFold, just like made such an advance, the experts were like, and changed the field. So now you have a DNA sequence, you can find the structure. Now, what's emerging is that you can also find the small molecule that binds it. And where it binds it, whether it activates it, where it binds else in all the human proteome. So this kind of acceleration. Toxicity, will it be toxic in the human body? Will it cross the blood-brain barrier? Will it have pharmacokinetics or not? All this stuff is AI food, right? And clinical trials is also something where it's already being done. Clinical trials, you optimize who the people are that are most likely to respond to the drug according to their medical files and history. So you can optimize to get the best statistical power on this very expensive thing. So my prediction is that in 5 years, there'll be a great acceleration in the ability to try out new drugs, new targets. So, in biology, there's converging technologies like cost of DNA sequencing, as you know, used to be many billions of dollars per human genome. Now it's like $1 going down. So it's huge power law like that. And the miniaturization, things that when I was a postdoc took a PhD to look at one protein interacting with another. Now you can do in 1 week all the proteins in the cells interacting with each other. So it's a scale up of 10 to the 4 and scale down in time of from 4 years to 4 days. And this stuff, the AI stuff. So I think what was in biology is going to be different. It'll be scaling up like engineering a little bit. So we're like on the transition now between, and this leads to some ideas you might think is ridiculous or maybe hopeful. One of them is called longevity escape velocity. That's the idea. So right now, every day, we gain 6 hours of life, right? That's not enough to live forever. But maybe technology will advance and each day will gain 8 hours. 10 hours. Well, maybe there'll be a point where each day will gain a day. That's the escape velocity. Okay? Blood-borne factors, so vampires can live forever by eating the blood of their victims, right? Not really forever. Did you see David Bowie kind of turn to dust in the movie Hunger? So there's actually serious research on, I told you about parabiosis, young mouse, old mouse, the old mouse rejuvenates. Exercise mimetics. You take a mouse running on a wheel, you find what factor in the blood, you can transfer it to an old mouse that's sitting as a couch potato and improve them. So there's exercise mimetics and also starvation mimetics or caloric restriction. So basically, it's like you get the gain without the pain. You don't need to diet, you don't need to exercise. So those kind of companies do that. Of course, there's also unethical companies that will sell you young plasma for $8,000 or you hook up to a young person, that's not real. Gene editing. So there's the power to, again, in the last 20 years, to change letters in the human DNA, or to do gene therapy, which is to take a virus and put new genes inside your body that can save people with rare genetic diseases by adding the gene that's mutated, like cystic fibrosis. There's a clinical trial actually going on people with Alzheimer's. They have the bad gene ApoE4 from both parents, as you remember, 12 times higher risk, and they get early onset Alzheimer's. So they're going to get Alzheimer's. They have Alzheimer's. And they try to, with gene therapy, put in ApoE2, the protective variant, in humans. That's the whale clinical trial. So there's some thought of doing that also. Gene, and maybe some doctors think, like Dr. studying leukemia says, why don't we take the embryo and remove that nasty mutation that gives you translocations to give you leukemia? We can change the DNA, no more leukemia. Is that worth it in editing? So that's an ethical problem. Where do you stop with gene editing? Even more science fiction, do you want? There's a line of thinking that eventually we'll be able to download our consciousness, our mind, onto a software. It's called mind uploading. The idea is that you create a kind of a, this is like the question about identity, right? Would you like to die and have a computer be you? What if there's a continuous process where we kind of make neural links with a buddy that's like us, stores more and more of our emotions and feelings and something, and they continuously, can you imagine something like that? Or somebody dies and you have an AI that completely, you put in all their longevity classes videos and you recapitulate me when I'm gone, right? The best me, like singing songs and stuff. You never miss me anymore. You can. All these kind of ideas have been there for a long time. Because of this AI and these neural interfaces, people are more thinking about it and talking about them. I think it's really science fiction. There's unbridgeable kind of, we don't understand how to bridge the gaps about the identity, the ethics, the many, many things I wrote down in the lecture notes, but I just wanted you to know that's an option for immortality until there's like a reboot and you have to upgrade and I want to talk a little bit about money. So another thing that's rising exponentially is the amount of investment. So in 2000, the global investment was 1 billion. 2020, 10 billion. The reason is that governments are trying to fund this, but also these weird billionaires, crypto billionaires. They're very young, and they invest like Altos and others. There's Calico. There's like each billionaire has his aging company, and also trillionaires, like our neighbor, the king of Saudi Arabia, has a fund that just invests $1 billion a year in research on aging. Nice, right? So the amount of biotech's also going like this. Well, basically, right now, I think it's the bubble. It's the second bubble. There was a bubble already with resveratrol and all kinds of stuff. Now it's, I think, the second bubble. I was alive in the .com 2000 bubble, where people said, one day all of commerce will be in the internet. And I said, what are you talking about? Like, I couldn't imagine it. Like, you go to the store, you buy something. There was a bubble. You can go to America almost without a visa and work. If you know how to program, then it just collapsed. But then 10 years later, Amazon, all the retail was on the internet. Right now it's obvious, right? You can buy things on the internet. It wasn't obvious in 2000, at least not to me. So there's these bubbles, and usually they're 10 years before. Like, about like self-driving cars bubble. So it's like, I think it's a bubble now, but I think there's also some science there that's interesting, that has potential. And for me, it's the beauty of figuring things out, connecting the dots, and having testable hypotheses that I think it's the best intellectual adventure out there. Next class, open questions. I know some of you have exams, you don't have to come, but if you want to discuss open questions in this field, I'll be here. For those of you watching at home, I hope you enjoyed, and all of you too. I really enjoyed it. I'm so sad that it's over. It's the highlight of my week. I love it. So maybe we should do the theme song. Aging, aging, aging here and there. Aging, 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 aging everywhere. Let's take a nice deep sigh of relief. Hope to see you on the road to great research, whatever you guys do. Thank you very much for coming to the course. We are officially over.